Good afternoon, everyone. We are group 4.6, and our topic is understanding evolutionary mutations of SARS-CoV-2 from genomic sequences. New coronavirus continue to mutate, and the mutant strains are emerging. Two strains, Delta and Lambda, identified in 2020, have become the dominant strains in the global epidemic. And now there's even a new variant, Omicron, spreading rapidly in Africa. Tracking and understanding the evolutionary mutations and the science behind them is important to vaccine and medicine development. In our project, we conclude these 5 objectives for elaboration.
Let me introduce the Objective 1 model of mutation rate. In genetics, the mutation rate is the frequency of new mutations in a single gene or organism over time. In our analysis, because the length of one nucleotide is very long, we view the organism as a unit, which means one case in the dataset. My statistic methods are mainly the descriptive analytics. The frequency of mutations is represented by the average number of mutated positions in the case per month. I compare each nucleotide with the reference and visualize the final results by line charts. Let's move to the findings part. I counted the average number of mutated positions in the case per month as a mutation rate in the world level and make a line chart. As the chart reviews, the mutation rate increased as time passed from around 3 mutated positions per case in January to around 11 per case in June. We could further see its change more intuitively from a fitting line chart. Roughly, there is a straight line where scope is 1.75 and y-intercept is 1. Considering the point mutations, CT mutations is the most advantageous mutation that takes more than 50%. We could see it as a favorable change for the virus. In further analysis, I decided to focus on individual countries. After data processing, we pick 4 representational countries, which are the USA, China, India, and Britain. Generally, the mutation rates in the USA and Britain increase faster than other countries. If we view the scope of the fitting line in world level as the average growth rate of mutation rates, we could see these 2 countries, they both exceeded. Next, our group mate will further discuss on the advantageous position of mutations.
With an overall understanding of the mutation rate, let's look at each position of the gene and check whether some positions are advantageous for mutation. The definition is that a position is advantageous if the nucleotides on the position are mutated in many cases and new nucleotides survives over time. So I count the number of mutated nucleotides for each position, and then I pick the top 20 positions as advantageous ones. And here is the result. Next, I check if the new nucleotide on each position decreases or even disappears over time, and I use a line chart to represent the percentage of nucleotides over each half-month period. In this graph, you can see that on position 3037, the new nucleotide T increases over time, so this is an advantageous position for mutation. However, in this example, which is position 28144, the new nucleotide C decreases over time and disappeared in May 2020, so this may not be an advantageous position. After checking each of the 20 positions, the final result is printed on the left.
So here comes to our objective 3, which is finding the advantageous pairs of mutation. During the mutation process, there are some positions that have a high probability of mutating simultaneously together. So we may define them as a pair of advantageous positions. So these are some examples. These 3 positions, we can see that they have almost the same pattern of mutation. We may predict that they are likely to become an advantageous group of mutation. So we use the process, or we use the method of hypothesis testing to check if 2 positions are correlated to each other. So we take the advantageous positions from objective 2, which is the 20 advantageous positions which have the highest number of the mutation time. And we check if every 2 of the positions are correlated. So here are our results. we sorted the p-value and find the top 10 advantageous pairs. After observing the pairs, we can form in the group, like the first 3 pairs we can form into the group 1. So here are our results. In objective 1, 2, 3, we are mainly focusing on the characteristic of the data. So in objective 4 and 5, we will be focusing on the association between data and search for more results.
Then follows the objective 4, which is to explore the association of mutation rate with time and location. In this objective, we want to look into and figure out 2 problems. One is how the mutation rate is related to the location. The other is the association between the mutation rate and time. The major researching methods that we adopted are linear regression and hypothesis testing. First of all, in order to better conduct the analysis, we need to do some data preprocessing to quantify the predictors that we want to analyze. To analyze the implication of location, we attempted to use the precise latitudes and longitudes to represent different locations. Since only cities in China were specified in a given dataset, we therefore restrict the location within China to do the analysis. Next is to quantify the factor of time. We adopted the months of all the records to represent the time. After proper manipulation of the data, we put the 3 predictors in columns respectively and add them onto the data frame. The picture on the bottom right shows what the data frame looks like after processing. Now that we've done with the data preprocessing, we proceed with simple linear regressions, assuming that both predictors are independent. We'll adopt the linear regression of latitude and mutation rate for illustration. As we may observe from the results, the slopes of both simple regressions are close to 0, indicating that location barely has any explicit effects on the mutation rate. But if we take a closer look at the summary table, where the p-value in the hypothesis is less than 5%, implying that the null hypothesis gets rejected. In other words, the predictor of latitude is statistically significant. To deal with the contradiction, one possible explanation is that the predictors aren't independent. For further verification, multiple linear regression might help. By repeating the similar procedure with extra predictor, we obtained a summary consisting of 3 predictors in total. From the summary table, we found that the p-values of all the 3 predictors have exceeded 5%, which verified the null hypothesis. Hence, it is highly likely that the predictors are dependent upon each other. Therefore, we may come to the conclusion that the location and the time are generally not associated with the mutation rate of the viruses. Thank you.
Next, we talk about objective 5, analyze the pattern of a mutation similar across different countries and locations. We use descriptive analysis and line chart to explore this objective. To define the pattern of mutation, we use the number of cases in total across the countries and locations as the indicators. Considering the high variation of sample size and different regions, we'll calculate the proportion of 12 mutations type instead of actual number to acquire the different degrees of substitutions. We rank the countries and cities according to the number of mutations and select top 10 with the highest number of mutation as the representative objects. After analyzing, we found that the percentage of mutation types in total sample across locations are quite different. The standard deviation are great. Take the city transition as an example. The percentage varies from more than 60% to about 40% in mutation cases across different countries. However, the main trend or pattern in different locations is similar. In the direction of mutation, the city transition is the main mutation type, following RTC, GT, and GA transitions. The difference may be due to different medical response and environment, as well as epidemic prevention policies. But at a macro level, the overall trends are the same. For structural reasons, the more structurally similar the bases are, the more likely they are to interchange. According to our analysis results, we have the following recommendations for the future research directions.
After completing our research, we have made some recommendations. In our process of working on our objectives, we have some problems and raise some questions, which is listed below. In the future, we may further analyze the association between mutation rate and other factors like the location in more countries and infection rate. In addition, we may do some prediction using this project so that this project can really do some help reflecting on the reality. So this is the end of our presentation. Thank you for listening.